Welcome to the Wednesday afternoon lecture series. I'm Kelly Tenhagen, a senior investigator in the Developmental Glycobiology section at the National Institute of Dental and Craniofacial Research, and I'm also the chair of the NIH Women Scientists Advisors Committee. I'm very excited to introduce today's speaker, Dr. Cassandra Extavour, who was nominated for this lecture by both the NIH Women of Color Committee and the NIH Women Scientist Advisors. Our speaker today is Dr. Cassandra Extavour, and she's the Timken Professor of Organismic and Evolutionary Biology and of Molecular and Cellular Biology at Harvard and a newly named Howard Hughes Medical Institute Investigator. Her Harvard lab exploits the disciplines of molecular genetics, protein chemistry, and mathematical modeling to understand early embryonic development, the genes that control it, the evolutionary origins of these genes, and how their functions have changed over evolutionary time. In studying Drosophila and also other non-traditional model organisms, Dr. Extavour has uncovered novel mechanisms and general principles of embryonic germline and gonad morphogenesis with clear implications for understanding human infertility. She's also discovered ways that environmental pressures on reproduction rate help steer evolution. Dr. Extavour is a native of Toronto, where she attended the University of Toronto and went on to obtain an honors Bachelor of Science with a specialty in molecular genetics and molecular biology. She obtained her Ph.D. with Antonio Garcia Bellido at the Severo Ochoa Center for Molecular Biology at the Autonomous University of Madrid. And she performed postdoctoral work first with Michalis Averov at the Institute of Molecular Biology and Biotechnology in Crete, Greece, and subsequently with Michael Acom at the University of Cambridge. In 2007, she established her independent laboratory as an assistant professor in the Department of Organismic and Evolutionary Biology at Harvard, where she was promoted to associate professor in 2011 and to full professor in 2014. Dr. Extavour has received numerous honors and recognitions, including being selected as an HHMI investigator in 2021, as I mentioned earlier, and also receiving the Ellison Medical Foundation New Scholar in Aging Award. In 2020, she was named a Harvard College professor in recognition of her contributions to mentoring and teaching at Harvard. The title of Dr. Extavour's talk is Evolving Adaptive Reproductive Capacity, a Systems Approach Using Drosophila Models. Okay, that's changed a little bit, but that's okay. And Dr. Extavour, welcome to the NIH, and thank you so much for being with us.
Thank you so much, Kelly, for the introduction. And I want to thank everyone who was involved in the invitation for me to be here. It's been scheduled for a little while now, and I'm really excited that it can finally take place. And I think hopefully we're all relatively used to the virtual format, which I think actually has a lot of advantages in terms of including more and more people. So I'm excited to tell you today about some of the work that we've been doing in my lab, one particular line of research that's kind of grown and built and added many different layers over the course of the years that we've been working on it. And we hope in the future to add even more layers going forward. And so that's why I decided to sort of think about this as a systems approach, because we're trying to encompass as many different elements of the complex system of biological organisms as we can in our study. We can't do everything all at the same time, but we can do a little bit of everything if we keep working on it long enough. The view that you're seeing here on this title slide is the view. It's not one of our field sites. I would say it's a view that you can have on the way to one of our field sites to collect some of the organisms that we work with as we've begun to work with natural populations to sort of enhance our work on laboratory populations of Drosophila species of fruit flies. So, I'm interested in the genetic and epigenetic underpinnings of biological function and form and diversity. And I'm also interested in understanding how all of these systems evolved. And so when we consider the function of a gene or the behavior of a cell, we want to understand both how that thing is working in a living extant organism and also how the evolutionary process that has taken place before we could really observe it might have led to that characteristic. So when we think about evolution, which is heritable change over time, we know that we need three main things for evolution to happen. Really, we just need two, and the third is sort of a bonus. So we need variation. if everything is identical, there's no change to be made. And so evolution can't happen under completely static conditions with no variance. In terms of evolutionary change, we're interested in heritable change. That means we need these variations to be heritable. So that's a second key pillar. With just those two things alone, you can have different patterns of heritability of variation, and you can get, by definition, evolutionary change. Now, if any of these variations are also linked to variations in fitness, then you have the opportunity for one particular mechanism to operate on the evolutionary process, and that is natural selection. I'm particularly interested in the link between variation and fitness. What I mean by that is I'm very interested in traits whose variability definitely has a link to fitness, because these are traits that are sort of more obvious candidates for the potential for natural selection to be acting. Because of course we remember that not everything that's produced in evolution is produced by adaptive forces. There are many non-adaptive forces that are very important. But today I want to see if we can make a step towards considering something that we suspect is of adaptive importance because it impacts fitness. And so we're interested in traits that impact reproduction, offspring production, because that's a key element of fitness, because it's a key element of sort of the number of offspring one produces is going to have a direct impact on the number of offspring in the next generation that are going to contain particular variants or allelic variations. And so we consider often sort of many aspects of the reproductive process. Since I sort of started as a developmental biologist, that's what made me interested in science as a profession. I think of all life as a developmental cycle, all cellular life. And then one of the definitions of life, of course, is reproduction. So for multicellular animal systems, which is what I'm going to talk about today, the reproductive process needs to involve, at the very beginning of development, a separation of cells whose DNA will be passed on directly to the next generation. So the cells that are going to make gametes, for example, you need a germline soma distinction. You then need to generate a bunch of different somatic cell types. This is a sort of an invariant property of obligate multicellular organisms like humans or fruit flies. The development process I'm just using as a huge box within which all of these different cell types organize themselves into a functioning entity. And once you've accomplished that, you can achieve your reproductive output. That might be a high or a low reproductive output, and that might contribute to your higher or lower fitness. To give you a sense of how we like to think about these sorts of problems in my lab, before we get into the nitty-gritty of a specific example, I want to let you know that we like to think of the evolutionary process and its sort of manifestation today on many levels of biological organization. We think about genes, their sequences, we think about the products that they encode, RNAs, proteins, what have you. We think about the cells and how they respond to these different gene products, how the cells put themselves together into organ systems, and how these organ systems function within organisms. And increasingly in the future, I hope that we're going to move this to another level of sort of biological organization, which is considering organisms in their natural environments, where they're, of course, having many interactions with a lot of other organisms. We take a multimodel approach because if we want to understand the evolutionary process, by definition, we're talking about heritable change. So we can't just look at one organism. We have to look at a minimum, many generations of an organism, and even better from our point of view, many organisms. Because when we compare the similarities and differences between different organisms, we can begin to make educated guesses, which we like to call hypotheses as scientists, as to the evolutionary origins of the things that we're seeing today. So I'm going to be focusing in today's talk on work that we've done using Drosophila melanogaster as a laboratory model, and a number of other Drosophila species, both within the lab and in the wild. But in my lab, other people working on other projects that I won't talk about today also work on other models that some people like to call emerging models because they're nowhere near as well established or as manipulable in the laboratory as something like Drosophila melanogaster. Yet we find them very fruitful to investigate because they can offer us insights into new types of biology that the standard lab models will never show you. So that means we sometimes spend time developing new models from particular places in the evolutionary tree that we suspect have new insights to show us that we can't see if we're only focusing on the part of the tree we always look at. Because everything we do builds on the historical work of many centuries of scientists before us, we also take very seriously considering the historical literature where, you know, the centuries of scientists who came before us have collectively studied so many more organisms than we ever can in our lifetime. And we don't want to neglect that massive data set. And so we often take advantage of that literature in a big way. And sometimes, again, it's not going to enter into this talk very much, but Simulating organisms is a very useful way, just like simulating any process is a very useful way to understand the sort of limits of potential evolutionary variation, and to generate informed expectations as to what you might see in natural extant populations. And then finally, we're not an organism lab, organism-driven lab, as you've seen, and we're not a sort of level of biological organization-driven lab, and we're also not a technique-driven lab. And so we use whatever technique we think is going to provide the most insight into the level of organization that we're interested in. And so that means that we use different tools, genetics, we use field work, we use microscopy, modeling, biochemistry, whatever we think is going to allow us to shed some light on that particular element of biological organization that we're interested in a given moment. Sorry, I seem to be missing a slide here. I'm going to interrupt myself for a moment because this is actually an extremely important slide that I'm missing, which is the slide to tell you about all the important people who did the work. So the team of people that really built up this system, the core members of this team are here, and they were members, current and former, of my lab over many years. Andre Green was one of my first graduate students, together with Didem Sarakaya. They've gone on to better and brighter things than my lab, but they were foundational in establishing the key developmental parameters that control ovarial development in Drosophila, and that's what I'm going to talk about a lot today. Sam Church was a student who just left us recently for his postdoc, and he really pushed forward all the work on the natural populations that we're going to see, but that natural population work in Hawaii was really pioneered in our lab by Didem Sarakaya, who's now at 10x Genomics. So this is the team of people that really laid the foundations for almost all the data that I'm going to be sharing with you today over many years. Andre and Didem were two of my first graduate students, and they laid the foundations for understanding the developmental basis of the ovarian processes that we're interested in, which I'm going to go into more detail about shortly. Didem was the first one from our lab to go out to Hawaii and collect these flies in the wild and figure out how to do it with the help of our collaborators over there. And then Sam picked up the baton from her and pushed forward all of the Hawaiian Drosophila work that you're going to see towards the end of today's talk. And then Tarun and Leo were a great collaborative duo within the lab. Tarun is a Drosophila geneticist, and Leo is a sort of true systems biologist at heart. And together they work to analyze the outcomes of sort of thousands of different phenotypes in hundreds of different genetic backgrounds to provide us with some of the systems level gene regulatory information that we were able to get about this system. Okay, so the specific study system now, the specific traits that we're going to be talking about, trying to understand their evolutionary variation, the evolutionary origins of variation in these traits. We're going to use a specific aspect of reproductive anatomy of fruit flies, and that's the number of a type of ovarian structure called an ovariole. So here on the left-hand side, we have a schematic of a fruit fly. It has a bilaterally symmetrical ovary inside it, which is the little white thing at the bottom. If we take out one of these ovaries and look at it close up, we can see that unlike a mammalian ovary, which is typically a sort of a bag of disorganized cells inside, and the cells inside are at differing stages of oogenesis, but there's not really a lot of anatomical organization within that. But in insects, they are very organized about their ovaries. They take their ovaries and divide them into individual assembly lines where eggs are made, and these individual assembly lines are called ovarioles, and you can see that over on the right-hand side. So the ovariole is organized in the following way. At the anterior, which is at the top of the ovariole, we have some germline stem cells that are essentially continually producing sources of new oocytes. The oocytes are produced initially up at the top in the anterior, and they differentiate and become more and more mature as we move towards the posterior. Then finally, they're completely mature, they're ready to be activated and fertilized, and then they're laid out the bottom of the fly or the posterior. So this trait now, ovarioles and the number of ovarioles, is a very interesting trait because, as I'm going to show you in the next couple of slides, it displays all of these key features of evolution that we would like to see in a trait whose evolution may be adaptive. That means may confer specific fitness advantages in specific environments. So we're going to look at ovariation in ovariole number because we think that this provides a direct link to fitness. And I'm going to show you some of the evidence for that thought in a second. So does ovariole numbers satisfy these key pillars of evolutionary change? Is it variable? The answer is yes. As you'll see at the end of the slide, at the end of the talk, ovariole number across insects is extremely variable. I'm talking several orders of magnitude variable. But even just within the genus Drosophila, we can change it by a couple of orders of magnitude alone. Here, what you're seeing is a set of evolutionary relationships between 12 different species of the genus Drosophila, and their ovarial number are these numbers down here on the right. That's the total number of ovarials per female. We find that the ovarial number is bilaterally symmetrical, and so the number of ovarials in one ovary would be just half of one of these numbers. And you can see that there are some flies like Drosophila seychellii up here close to the top that has about half the number of ovarials total, as some of its very close relatives like Drosophila simulans or Drosophila melanogaster. So we've got a lot of variation to work with in this trait, so evolution looks promising. We have high heritability of this trait. This trait can be influenced by environmental parameters, as you'll see, 20 minutes or something. But let me tell you that it is very strongly heritable, very strongly genetically determined, and here's some evidence for that. In this experiment, what we did was go to Hawaii, find flies that naturally are living and therefore we think feeding on different types of substrates, flowers, sap, etc. They're listed at the top. And these different species, we catch these wild-caught females. We don't know what they've been eating, who they've been mating with, what they've been doing in their lives before we caught them, but we catch them, we bring them back to the lab, we give them standard fly food, melanogaster food to lay their eggs on. They lay their eggs in lab. Their eggs are happy to develop on our food, and their larvae are happy to develop and eat our food. And then what we can do is compare the ovarial number of these wild-caught mothers that were eating whatever they wanted in whatever environment they felt like. We can compare their ovarial number to the ovarial number of their lab-reared offspring. And we find no significant difference, even though all the moms were eating whatever they wanted, but all the offspring were eating the exact same thing in the same lab conditions. Nevertheless, their ovarial number between these two generations is not statistically significantly different. So this is telling us that we have a huge amount of genetic control over this trait. So that's 2 check marks so far for evolutionary change. Right away, we know we can get it in this trait. And finally, we think that ovarial number evolution or change over time may actually be adaptive in such situations, because in Drosophila, the more ovarials you have, the more eggs you get to lay. This experiment here, these data are showing you the results of an experiment where we used some genetic tricks in Drosophila melanogaster to make the flies have more and more ovarials. So if you look at the right-hand plot, our wild type sort of control strain is at the top, and it has a certain number of ovarials. And in the second row, you see that we've manipulated the flies genetically. The details are not so important, but we've increased its ovarial number. So this bar is shifting to the right. And correspondingly, if you look at the left-hand plot where we're looking at the total number of eggs laid by these females, they're laying more eggs than they did when they had fewer ovarials. And then we can do even more genetic tricks in the bottom row. We can give them an even higher ovarial number, and correspondingly, they're going to lay more eggs. Over here, you can see some more evidence a little further afield that ovarial number is very strongly positively correlated with egg laying in some insects, including in Drosophila. So our laboratory North American control strain of Drosophila melanogaster has about 40 ovarials per female. And in our hands, she's going to lay 50 to 100 eggs a day, give or take. This other species of insect that we also rear in the lab, this cricket gryllus, has an order of magnitude more ovarials, and it lays an order of magnitude more eggs. And even just within very closely related species, these two Drosophila species, melanogaster on the top and seychellii on the bottom, they shared a last common ancestor only about 3 million years ago. Seychellii, nevertheless, has only about half the number of ovarials compared to melanogaster, and it lays far fewer eggs than melanogaster. The x-axis is showing you the first 10 days of life, and what we did was count up the number of eggs that these females laid on every day of their first 10 days of life. That's the number of eggs that's on the y-axis. And you can see that at least for these first 10 days, these two flies lay very different numbers of eggs. Each line is an individual female that we counted. This was a small pilot data set where we just had 10 females per species, and we just counted for 10 days. But we've just finished collecting a larger data set with 100 females per species over their entire lifetime. We counted how many eggs they all laid until they died. And Seychellii never catches up. It starts off with fewer ovarials, it starts laying fewer eggs, and it can never lay more eggs than melanogaster. It's always going to lose out there. So we have this quantitative trait, highly heritable, linked to fitness. And now we're going to start to investigate a number of different questions about how this trait gets to be the way that it is in specific species, and how variations in this trait might possibly have evolved. So we're going to look at it from at least 3 different points of view. And I'm just going to give you examples of the sorts of experiments that we do when we're concerned with the developmental control of the trait, the genetic control of the trait, and the environmental control of the trait. So when we think about the developmental control of the trait, what I mean by that is, what are the cells that need to make the ovarioles? What do the cells do that makes them decide how many ovarioles they need to make? What you're seeing here is going to become clear shortly. It's a micrograph of a larval ovary before the adult ovary is formed, before the ovarioles are even finished. And you can see that there are a lot of different cell types. They're expressing different types of genes, the different colors. The genes aren't even important. What's important is that they're already beginning to organize themselves in a way that, as I'll show you, corresponds to the adult ovarial number that these larval ovaries are going to give rise to. So ovarial number, it turns out, it's an adult trait we're interested in, but it's determined during the larval phase in this insect. So during the embryo, the animal makes a primordial gonad, which is just a handful of germ cells in yellow, surrounded by a little ring of somatic cells in white. And they're, at this point, as far as we can tell, pretty undifferentiated, these somatic cells. As the larva grows, so the different L numbers are different larval instars or different larval stages. As the larva grows, all the cells proliferate. You get more germ cells, you get more somatic cells. The somatic cells reorganize themselves. So you can see all the somatic cells are up on the top now, the white cells, and the germ cells are all down at the bottom or the posterior, and a handful of cells have turned green. These are a special type of somatic cell, and they go and they hang out or they intermingle with the germ cells. So we call them the intermingled cells. So the somatic cells are moving physically, and they're also starting to become different in their identity and behavior. Then we get further differentiation of the somatic cells as we proceed in development. Some of the cells begin to not only express different genes than their neighbors, those are the red cells. Other cells, they're expressing other genes that I'm not showing more colors here, but they're starting to move down beside the germ cells, as you can see. So there's some cell migration happening. As development continues, these red expressing cells begin to change shape now. They become, instead of being round, they become kind of flat and disc-like, and they stack up on top of each other. The migrating cells on the right-hand side keep migrating. They're heading down to the posterior of the ovary. And finally, at what we call the LP transition, which is the larval pupal transition. So this is when the animal's about to head into making a pupa, which is the final stage before adulthood. You get the red cells, which I'm going to tell you are terminal filament cells. They've made these stacks called terminal filaments. Terminal filaments are critical because the number of terminal filaments that the animal has when it goes into pupariation is the number of ovarioles that it's going to have when it hatches out as an adult. And so that means that to understand quantitative variation in ovarial number in adult flies, what we really need to understand as developmental biologists is this cell behavior that takes place during larval stages in the ovary. Here are some micrographs to show you sort of what these things really look like to us under the microscope. These are different larval instars showing you the larval ovary. All the nuclei are in blue and the terminal filament cells are in red. As they begin to acquire their identity as terminal filament cells, they turn on a bunch of special genes, including this red gene. It's called engrailed for those of you who like to know gene names. The cells are changing their shape. They're becoming kind of flattened and disc-shaped. They stack up into little stacks that we've outlined with a white dotted line for you to see more clearly, with the result that by the time it goes into pupariation, this larval ovary now has the same number of terminal filament stacks as it will have ovarial numbers in the adult. Okay, so this is how in one species ovarioles get made, and how ovarial number gets determined is by determining the number and organization of terminal filament cells. What happens if we look at two or more species whose adult ovarial numbers are different, and we kind of go backwards in time and we try to find the point during larval development when the cells began to behave differently in the different species to give us the result that as an adult they have these different traits. We chose for this study to look at convergent evolution of lower ovarial number relative to an ancestor. So this is a phylogenetic tree showing the relationships of a number of different Drosophila species. The numbers on the right-hand side to the right of each species name are the number of ovarioles in one female of a living, sort of an extant adult female of this species. And the numbers in the yellow boxes are the numbers that we infer through an ancestral state reconstruction, the last common ancestor of these species might have had. And so we can see that if we focus down on the bottom of the tree here, Drosophila melanogaster, this is the guy that we know. OR means Oregon R, that's our wild type North American, collected in Oregon a while ago. Our Oregon R reference strain has about 36 ovarioles in a female. Another strain of melanogaster, it's the same species, but it lives in India, has a much lower ovarial number, about 24. And then if we look at not a different strain of the same species, but a different species entirely, Drosophila Seychellia down at the bottom, as you've heard before, it has many fewer ovarials than melanogaster. And what we're looking at here is what we think is the evolution independently in these two different lineages of a lower ovarial number compared to the last common ancestral number of this group, which we think was around 32 or something like that in the bright yellow box. So what Andre did here was he looked very carefully, he counted the number of cells, he documented their behavior in the larval development of the ovary of all of these three lineages, two species of melanogaster, one with high, one with low, and Seychellia with the low number. And he asked, do India and Seychellia have lower ovarial numbers than the other lineage for the same developmental reasons? Did the things that their cells did differently, are those things the same between those lineages, or are they different between those lineages? And the answer is they're different. In other words, there are many different ways that you can lower ovarial number. There are many different choices that cells can make, with the ultimate result that they'll have fewer ovarials than some relative. This is the larval ovary at the very beginning of larval development of these three lineages. India is on the left, Organar is in the middle, and Seychellia is on the right. Andre counted up the total number of somatic cells in that ovary at the very beginning of larval development and saw that Seychellia, which remember is going to have a very low ovarial number as an adult, indeed has a smaller number of somatic cells in that gonad primordium at the beginning of its development. The other two lineages, which are both the species Drosophila melanogaster, have actually the same number of somatic cells. That's interesting because remember, we're expecting India to grow up to have fewer ovarials, but when it starts making its gonad, it has the same number of somatic cells there. As development proceeds, all the cells are going to proliferate, we know this. So at the LP stage, when we're just heading into preparation, Andre counted up these cells again, and we found that, as we might expect, Seychellia has fewer cells in its ovary than Organar, than melanogaster for North America. Seychellia started with fewer cells. We know it's going to end up with fewer ovarials, and indeed, it wasn't able to catch up by proliferating more in the larval stage. It starts small, it stays small. But the India strain is very interesting because we know it's going to grow up to have fewer ovarials than its North American cousin. And yet, when it's time to make a pupa, this ovary actually has way more cells in it than Drosophila melanogaster does. And you can even just see that physically. It has this massive larval ovary. So why is it not making a lot of terminal filaments if it has all these cells? It turns out that it's just allocated these cells differently to terminal filament fate or to not terminal filament fate. You might remember that when we were looking at the cartoon of larval ovarian development, there's a bunch of cells, somatic cells, that start anterior to the germ cells, and they migrate down to the posterior. It turns out that group of cells is much bigger in India than it is in melanogaster. Can you still see my screen? It looks like somebody else has started screen sharing.
Indeed, that is the case. I don't know what's happening. Maybe you can reshare your screen.
Okay, I'll do that. How about now?
Yes, you're back. I'm sorry, I have no idea what that was.
No problem. So what India did, even though it started with the sort of regular sized ovary, it proliferated way more, but then it sent way more of the cells that it had to the posterior of the germ cells. That left a smaller proportion of cells in the anterior region, which is where we know the terminal filament cells have to come from. So this little study told us that there are a lot of different sort of cell decisions that you can make and that you can change over evolutionary time to end up with a different adult result, depending on the lineage that you're in. Now, one thing that Didem wanted to know when she went to Hawaii for the first time was if the key parameter that controls ovarial number in Drosophila melanogaster, which is terminal filament cell number, is that key parameter also a key parameter that correlates with variable ovarial number in natural populations? So to answer this question, she went to Hawaii, she collected a bunch of different species that all have very different ovarial numbers. So the adult ovarial numbers are on the plots on the bottom. And she counted up their terminal filament cell number in larval stages, which you can see in the gray plots on the top. And you can see there's an almost perfect one-to-one correlation between the number of terminal filaments that you make and the total number of ovarials you're going to have, even in these wild populations. So this suggestion we were getting from our lab species, melanogaster, that terminal filament cell number is the key cell biology parameter to control ovarial number, this observation seems to hold up for these natural populations. Okay. Let me tell you about some of the ways that we look at the genetic regulation of ovarial number. How do we figure out what genes control the number of ovarials and how those genes and their activities might be changing in different species to give us different results at the end? This is a little schematic, one of these hideous signaling diagrams that just shows you some of the different signaling pathways that were known to be involved in setting up the correct number and orientation and arrangement of these different key cell types in the ovary, I don't know, 10 years ago or something when we started this work. So terminal filament cells in red, intermingled cells and germ cells. The numbers and placement of these are all really important to get your correct ovarial number. Dita did a nice study where she showed that hippo signaling, which is a pretty recently discovered conserved proliferation controller signaling pathway in animals, also plays a key role in this system by controlling the number of terminal filament cells. Now we know, as all developmental biologists know, that there's no single gene that controls any single trait. So we took advantage of the new knowledge that we had that hippo signaling was involved in regulating terminal filament cell number. And we asked what other genes might also alter ovarial number together with hippo or independently of hippo. And so for the fly nerds in the audience, we did a classic sort of suppression screen, enhancer screen. We did a modifier screen in a hippo loss of function background. And then we systematically knocked out using RNAi every one of the members of all of the animal conserved signaling pathways. So about 800 genes one at a time. We knocked them out in a wild type background and in a hippo loss of function background. And we asked if this affected the ovarial number of the flies and if it affected their egg laying. So we had two different reproductive traits we were looking at here. And if you are working on a signaling pathway and you're wondering if your signaling pathway was involved in ovarial number determination, the answer is yes, because every conserved animal signaling pathway is involved in ovarial number determination, it turns out. One of the ways, though, that we chose to look at the results of this screen, which gave us several hundred new genes involved in ovarial number determination, was to look at it from a systems biology lens through the perspective of asking what the relationships were between the genes that we'd realized are important for ovarial number and their protein-protein interactions, which might suggest some kind of gene regulatory relationship. So Leo took the known protein-protein interactions from the entire Drosophila genome and kind of superimposed that onto the results of our functional screen here for the different phenotypes that we'd looked at, egg laying and ovarial number in a hippo loss of function background or in a wild type background. And what he discovered was that there are certain genes that interact with each other more often than you would expect by chance, that also impact the same phenotype more often than you would expect by chance. These more often than you would expect by chance relationships suggest to us that these might be conserved functional gene regulatory networks, that genes that work together to control these specific traits. We found genes that work together to control both of the traits that we looked at. They shut down ovarial number and egg laying, or they raise ovarial number and egg laying together, sort of all or nothing group of genes. And we also found sort of what we're calling submodules here, which were smaller groups of genes that together appear to regulate just one element or just sort of like one leg of the phenotype. Like this group of genes up here at the top, it regulates ovarial number only in a hippo loss of function background, these groups of genes here, and so on for these different phenotypes that we'd scored. The most exciting part of this screen here was not to discover that some of the genes that we tested for function actually function in ovarial number. I mean, we knew we were going to get something. But what was exciting about this topology analysis was that because it kept in mind all known protein-protein interactions from the entire Drosophila genome, not just the 800 genes that we screened, but all of them, it was able to predict for us what we call here connector genes in this topology analysis, what I've marked in the blue circles. These are genes that we hadn't actually tested in our screen, but according to the network analysis we did, they should be involved in very specific phenotypes, because you can see that some dots are blue and some submodules and some aren't. And then what we did was we tested all the blue genes. We tested all these genes that the network predicted should be involved in our phenotype based on what we had observed. And we found that actually even more often than in our original screen, these network predicted connector genes were also key regulators of ovarial number and melanogaster. Okay, so we have a lot of information about a lot of genes, right? Everybody can say this. Is there a way that we can identify if there are some of these genes whose activity is different, has evolved to be different between species, that might explain to us why these species end up with different ovarial numbers? Here's one example of one of these types of studies that we did. For a number of different reasons, we suspected that insulin signaling was very important to regulate ovarial number in Drosophila melanogaster. You can raise insulin signaling levels with gain of function mutations and you'll get more ovarials and more eggs. And you can lower ovarial number by depressing insulin signaling and you'll get fewer ovarials and fewer eggs. Since we knew that Seychellia had fewer ovarials than melanogaster, we asked if there was sort of a systemic evolved difference in the level of insulin signaling in the ovary of Drosophila melanogaster versus Drosophila seychellea at this key time in larval development when they're making their ovarial number. We found the answer was yes. Here's one way that we looked at that. We want to compare the levels of insulin signaling between two species that we know have different traits. One way that we did this was to quantify the amount of phosphorylated AKT, which is a downstream effector of insulin signaling in animals. And we found that the levels of phosphorylated AKT, which is a proxy for sort of insulin signaling levels, were higher in Drosophila melanogaster with more ovarials than they were in Seychellea with lower ovarials. And through a number of other experiments that I'm not going to show here, we found evidence that the Drosophila seychellea insulin receptor allele has actually evolved a lower level of activity compared to the melanogaster allele. And that in part accounts for the Seychellea lower ovarial number that it's also evolved in the last 3 million years. All right, so for the last bit of the talk, what I want to turn to are some of the ways that we're considering environmental regulation of this trait. I've talked a lot about genes and genetic control of cell behavior, and I've implied that it's a very heritable trait, and indeed it is. That doesn't mean that it's completely impervious to environmental regulation. Let me show you here one example of what I mean. We know from the work of previous researchers that starvation, which remember is mediated by insulin signaling now, that starvation has a big impact on ovarial number. If you starve your flies, they have fewer ovarians. Okay. Those of you who work with flies or work with any animal, honestly, probably have observed that if you starve your animals, they're going to be smaller. They're probably going to have less cells total, and the cells they do have are probably going to be physically smaller. And we did some measurements of cell numbers and cell size, and indeed the starved melanogaster flies had fewer smaller cells in total, including fewer terminal filament cells, and that's what explains how come they get fewer ovarials when you starve them. Now, we also noticed, and this starvation effect, by the way, applies to all the species that we've studied. So you starve melanogaster, fewer ovarials in number, fewer ovarials than normal. You starve Seychellea, fewer ovarials than normal. But here's the interesting thing that we noticed. The degree of reduction in ovarials is much greater in melanogaster than it is in Seychellea. So here what you're seeing, the black bar is melanogaster, the white bar is Seychellea, and the red bars are these two species, melanogaster on the left, Seychellea on the right, their ovarial numbers on the y-axis when you starve these flies. So when you starve melanogaster, the right pair of bars, the flies get about 20%, almost 20% fewer ovarials is what they end up with. When you starve Seychellea, they only end up with about 10% fewer ovarials. I've told you that we have evidence that insulin signaling systemically is operating at a lower level. It's evolved a lower level of activity in Seychellea compared to melanogaster. And we suspected that perhaps because this key nutrient sensing pathway is operating at a lower level, Drosophila seychellea is sort of less able or less apt to detect changes in nutrition when we subject it to starvation, with the result that it notices that it's being starved, its ovarial number notices and drops accordingly, but not as much as melanogaster. If this is the case, then we might expect that different populations of Drosophila seychellea, maybe living in different parts of the Seychelles, which is where this species is endemic to, they might display lower degrees of sort of environmental or population level variation in their ovarial number compared to Drosophila melanogaster, that we hypothesize that different populations of melanogaster from around the world, because of their higher levels of insulin signaling and their greater ability, therefore, to detect nutritional changes, we might observe a greater variance in ovarial number when we compare different populations of melanogaster from around the world. So Andre got his hands on a bunch of different populations of melanogaster from around the world and a bunch of different populations of Seychellea from around the Seychelles. He compared their mean ovarial number, and the key thing we're asking about here is the variance. And we found that our prediction was supported by the data that we gathered. So the black symbols, white and black, are the ovarial numbers on the y-axis of different Drosophila melanogaster populations from all over the place. And then the red symbols are the ovarial numbers, the mean ovarial numbers of Drosophila seychellea populations from around the place. And that Seychellea variance is much smaller than the melanogaster variance. So our hypothesis here is that because the molecular mechanism is the same, both for nutrient sensing and for heritable change in ovarial number, this evolved difference in this key nutrient sensing signaling pathway might explain not just species-specific differences in ovarial number, but species-specific degrees of phenotypic plasticity in ovarial number, which is what we're seeing here in these populations. All right, so to end up this talk, I want to tell you about some of the work that we're doing in Hawaii and how this is helping us think about this problem. We've looked at a range of ovarial numbers from something like 10 to 20 per ovariole by doing these Melanogaster comparisons, right? And that's kind of as much as we can push it in this group of 12 Drosophila species that I showed you at the beginning. These are just the 12 Drosophila species whose genomes were first sequenced and made publicly available. There are about 4,000 described Drosophila species worldwide. About 1,000 of them evolved only about 20 million years ago on the Hawaiian Islands. This group of Hawaiian Drosophila, it's about a quarter of all worldwide Drosophila diversity. It is incredibly variable in terms of all sorts of morphological and behavioral traits, including ovarial number, much more so than the other approximately 3,000 Drosophila species from the rest of the world. The continental ovarial number range, as we've talked about, is sort of between 10 and 20 per ovariole, but in Hawaii, you can find Hawaiian Drosophila species with only 1 or 2 ovarioles in their ovaries, or almost 100 ovarioles in their ovaries. And so we're really interested in understanding the genetic and ecological basis of this huge variation in this group of species that underwent an explosive island adaptive radiation in a very short period of time. So to do this work, it involves field work. It's not very glamorous. You're literally sitting on the forest floor in a rainforest, digging through mud and rotting vegetation, looking for maggots. It's not amazing, but the insights that we hope it will give us are pretty amazing. So we go to Hawaii, we identify these key forests filled with endemic Hawaiian plants, because as you'll see shortly, these Hawaiian Drosophila have evolved very specific associations with very specific types of plants that are also endemic to the Hawaiian Islands. If you find some maggots, you might get lucky, but more often we catch the adults and we literally have to hand catch them one by one with a series of different sneaky tricks and traps. And some of these species are able to come back to the lab and live with us. And I want to say that we could never have accomplished this work without our key collaborators in Hawaii, Ken Kaneshiro, Steve Montgomery, and Carl Maniaka and Don Price were all critical, kind collaborators who helped show us the islands, show us the plants, and introduce us to the flies and help us rear them. So I need to thank them as well. So I've already said that Hawaiian Drosophila make up a huge amount of worldwide Drosophila diversity, and we can also, through a combination of basically a biogeography phylogenomic study, we know, and others before us have also looked at this, the approximate origin in time and in space of these different lineages, sort of who originated where. And the short version of the story is that many lineages arose on many different islands independently, and there are also many lineages that traveled back and forth between islands, okay? I've mentioned already that the Hawaiian Drosophila really have specific preferences for endemic Hawaiian plants. So some of them are much more specialized than others. Some of them will only lay their eggs and on certain fruits or on certain fungi or in the spider cocoons of a specific endemic spider species in Hawaii. Some of them are really specific. And some of them are more generalist, like any rotting bark of the following 5 plants, they're good to lay their eggs there. Where they lay their eggs, we think is key, because where they lay their eggs is where the larvae will hatch. And where the larvae will hatch, that's what the larvae will eat. And as you've already seen, there's a great deal of nutritional plasticity potentially available to contribute to evolution in this system. And so what you're seeing here is a tree of a number of different Hawaiian Drosophila species. They fall into 5 major groups, which you can see in these little vertical clades here, the Scaptomyza, which are the outgroup to all of the rest of the Hawaiian Drosophila, the so-called modified mouthparts, MM, the Haleakala, which feed exclusively on fungi. You can see all the Haleakala here have yellow symbols. They only lay their eggs on fungi. They're a very understudied group. They're hard to catch. We have the so-called AMC group, and the AMC and the MMs, you can say they really like these green substrates. They've evolved a great degree of specialization on the leaves of specific Hawaiian plants. And then we have the so-called PNA clade, also known as the picture wing clade, and they are often bark breeders with a few different variations. And the circles on these, on the little different nodes and lines of this tree, indicate places where our analyses suggest that there have been evolutionary shifts in host preference. So all of a sudden, a lineage will evolve a different preference for a substrate to lay their eggs relative to its ancestors. So with all of this information combined, we have a little bit of information about the evolutionary relationships of the flies, where they live, their ovariole numbers, their substrate preferences. Ultimately, what I'd love to get at are what are the evolutionary differences in gene expression and function between these different lineages that can help us explain the evolution of different genetic control of these variable traits. Here we're talking about ovarian number. Now you might say, okay, well, you know about the genes in melanogaster. Why don't you just, sequence a transcriptome or something of these Hawaiian fly ovaries? Then you can compare them and see which genes are expressed differently, and then those might be the genes that are going to be important. And that is absolutely a great approach. However, one thing that we have to think about is, okay, we're going to sequence, let's say, the transcriptomes, the genomes, whatever you feel like sequencing between these different species. And you're going to say, okay, well, if I see differences, then maybe those are the differences that are responsible for the trait difference between the species. Here's the thing. There's going to be differences. These are different species. They've evolved to be very different over, you can quibble about what's long and what's short, but we expect them to be different, right? I was saying earlier to Susan, why don't I have an elephant's trunk? Well, I'm not an elephant. We shared a last common ancestor a little while ago. So we've evolved to be very different. Our question is, how can we tell which differences are greater than those that we would expect to have occurred just because these lineages have been diverging? Which are the gene expression or gene function differences that we should really be paying attention to, because they might explain this adaptive evolution of different ovarian members. So I want to take you just for a couple of minutes through a little thought exercise to illustrate the complexity of this problem. Let's say that you're just doing a 3-species comparison. You've got 3 species of your favorite insects here, and you're comparing the timing of gene expression. And you find that in 2 species, your gene that you like is expressed at one stage, and in the other species is expressed at a different stage. You might say, well, by sort of strict parsimony, I've got 2 species expressing at stage 1 and only 1 species expressing at stage 2. And also, one of my species that expresses at stage 1 is the outgroup to the other 2 species. By strict parsimony, I'm going to hypothesize that the gray expression state, so the stage 1, is the ancestral state, and then the blue is the derived state. And you might say, okay, well, this seems reasonable. I've certainly read lots of Vivo Depot papers, including our own, that make this sort of inference. Okay, here's the thing. When we think about the evolutionary process, we have to admit that things could be a lot more complicated. Because we know that there are not just 3 insects in the world, nor have there always only been 3 insects. There have been many more insects extinct than the 1 million insects that are described that we know are alive today. And collectively, the course of evolution of these insects spans literally billions of years of evolutionary time. So there have been billions of opportunities to make evolutionary change. Now you could say, well, in my very limited taxon sampling, I sampled 3 species out of 1 million living species. Maybe there was literally only 1 evolutionary change in this trait, the blue arrow, and I just happened to catch it by chance. That could be, but it seems a little less likely. Then there wasn't only 1 change, but you happened to catch one of the changes. And the question is, how many changes are there? And how can we know when we're only sampling a very small amount of the living variation? And we can't observe the previous variation, and we know we haven't even observed all the living variation. So you might say, well, I don't think this trait changes very quickly over evolutionary time. So I don't think that there were that many changes. There were more than 1, but I don't think there were that many. But I did get lucky, and I sampled one of the very few that there are. But what if this trait changes very quickly over evolutionary time? What if it's contributed to by 300 genes, all of which are potential targets of evolutionary change? So you might expect a great degree of evolutionary change. What if mutation rate is particularly high in this organism, or high at the hotspots of the genome that contain the genes you care about? Now you start to think, can I really be very confident that the blue state really is a derived state and not an ancestral state? And of course, we also know, we know so little about the evolutionary process when it comes to these deep time events that we really have to be honest and say we can't exclude the possibility that we literally can't say anything when we do a taxon sampling of 3. Okay, so how are we gonna deal with this problem here in the Hawaiian Drosophila? Here's how we're beginning to think about it in the last couple of slides. Sam went to Hawaii, collected 12 different species of Drosophila, which are in the gray boxes over here, and sampled 12 of them here in the cyan little things for expression analysis. These wild-caught animals, he created sort of bulk-seq RNA transcriptomes of their heads, because we're interested in some nervous system function for behavioral reasons, of their ovaries, and then a transcriptome of the rest of the carcass. He also sampled a transcriptome from a single whole individual from each species, and these are all individual RNAi, so from sequences, so from 1 ovary, from 1 head, et cetera, per species, in triplicate, in biological triplicate and technical triplicate. And what we're asking is, What are the expression differences between, we're going to focus on the ovary, between these different species. We know there's going to be differences, but what are the differences that we should focus on? Here's a plot of the sort of expression bias. So on the right are genes that have higher expression in the ovary than in the rest of the body. On the left are genes that have higher expression in the whole rest of the body rather than the ovary. And we asked which of these genes are known to be highly expressed in the Drosophila melanogaster ovary, because those are likely to be functional, at least in the melanogaster ovary. And are there potentially sort of ovary biased genes that have evolved different degrees of bias in different lineages of the Hawaiian flies? Okay, so we already know, and this plot is showing you, there's lots of evolved changes in gene expression, even just in this ovary. Now we can say, okay, are there genes that have evolved this ovary biased expression relative to some of their neighbors or some of their cousins? We know the evolutionary relationships between these flies, and this is a critical prerequisite for doing this sort of analysis. So then we can say, okay, in this species, not only is this gene biased towards expression in the ovary, it's got more expression in the ovary than the rest of the body, but it evolved that newly from an ancestor that didn't have that sort of bias. Those are the red dots. We can also ask the other sorts of question, which are genes that didn't used to be biased in expression in the rest of the body, in the non-ovary, but then they evolved that carcass bias. Those are the blue dots and so on. So again, we have lots of changes. We can pinpoint the sort of evolutionary direction of some of these changes, but which ones of these changes are kind of unusually large or unusually interesting to us against the backdrop of lineage diversification that we know is already happening? So we can ask a couple of questions. We can say, are there groups of genes that evolved an especially large change in expression bias? Those might be interesting candidates relative to the whole transcriptional activity background of the fly. And another question related is, we can ask, are there groups of genes whose expression evolves in the same direction? We all evolve an ovary bias. Maybe these are genes, again, that evolve an ovary bias together in particular lineages, but not as a sort of simple consequence of the evolutionary diversification of these lineages. These might be changes we're interested in. And so Sam developed a couple of methods to ask these two different questions. In this plot here, what we're doing is we're basically asking how much of the variation in ovary biased expression that we see between different lineages, these are clades A, B, C, and D, how much of that different expression appears to be attributable to just the evolutionary distance between the lineages, which is on the y-axis, which is on the x-axis, and how much of it might be due not to there's been a lot of diversification between these lineages, but there's been a lot of change between these organs. When we ask the question that way, it helps us see what should be the next steps we should take in this analysis. We should choose, for example, genes that have evolved a very different degree of ovary bias in flies from clades A and B, for example, because their ovary expression is more different than we would expect, given how closely related they are. Choosing, for example, to compare a species from group A with a species from group D and saying, whoa, I found some genes that are really differently expressed in the ovaries of species from A and species from D, that might not be such a great choice for our next experiments, because we already expect those genes to be really different in their degree of bias, given the amount of diversification that's happened between these lineages. And then finally, another way that we can think about this problem is to leverage information from Drosophila melanogaster, where pretty much every genetic interaction has been mapped. And we can ask if there are groups of genes that are known to interact with each other in Drosophila melanogaster in functionally relevant ways like physical protein-protein interactions on the left-hand plot, or genetic interactions of enhancement or suppression in the right-hand plot. And we can ask whether genes that in melanogaster interact in these known ways are more likely to evolve concerted ovary bias or loss of ovary bias in the Hawaiian Drosophila. And indeed, we find that genes that evolve similar biased expression in this organ or that organ, we care about the ovary here, they do tend, according to the melanogaster data set, to have some sort of functional relationship with each other, whether genetic or physical. So these types of approaches of these comparative transcriptomes that we've got now give us new testable gene regulatory hypotheses that we can go in and investigate in specific, sort of logically selected Hawaiian species to try to understand what genes have evolved to give us differences in expression that help us explain the lineage-specific diversification of ovarial number, which may have been adapted on these different substrates that they evolved their special relationships with. And then finally, I want to leave you, having spent so much time on melanogaster, let's zoom out on Drosophila. Let's zoom out for a second and consider all insects. We've said that in the genus Drosophila, we have a range of ovarial numbers of 1 to 100, give or take. That ovarial number is very strongly correlated with egg laying. It turns out neither of those things are true if we zoom out and look across all the insects. So what Sam did was look through literally a couple thousand different papers from the literature that other people have counted ovarial number before us. So we don't have to count all the ovarial numbers in the world. We can look at what others have already done. And we can plot their ovarial number on the x-axis here. And here we're just grouping on the y-axis sort of different major groups of insects. The colors are just meaningless, so you can differentiate them from each other. And we can already see that there are insects that only have one ovariole in their whole body. This means that they do not have a bilaterally symmetrical ovary, okay, as I was telling you, Drosophila does. And there are insects that have more than 10,000 ovarioles in their body, okay. These, spoiler alert, tend to be the social insects, like ant queens and so on. Still though, even if we discard the social insects, which have their own thing going on, we've got some beetles that have over 1,000 ovarioles in an ovary, okay. So there's a massive amount of variation. I don't know if variation in terminal filament cell number or behavior can explain all of this variation, right? Even more crazy to me is the fact that we observe repeatedly groups of insects that have evolved an invariant ovarial number. If you remember the melanogaster-Seychella comparison I told you, where we looked at the degree of variance in ovarial number in different melanogaster populations from around the world or different Seychella populations from around the Seychelles. And I told you, I kept saying ovarial number, mean ovarial number. There's a species-specific mean ovarial number in Drosophila. In all Drosophila species that we've looked at, if you dissect 100 flies, they don't all have exactly 10 ovarioles. They have 10, plus or minus 2, depending on the species. However, there are many groups of insects, and you can sort of see them intuitively in these little vertical lines that pop up here and there towards the left-hand side of this plot. These vertical lines of dots. These are insects that have absolutely invariant ovarial number. You can dissect 100 and you'll always find the exact same number of ovarioles in there. I also don't think that the cell biological and developmental morphogenetic programs that we've discovered are important for ovarial morphogenesis and number determination in melanogaster. I don't think those programs can explain how you always get the exact same number of ovarioles every time. And yet, this is a common occurrence that's evolved independently many different times in the insects. So going forward, another direction that I'd love to go in is to bring some other insects into the lab that have these other unusual patterns. They've only got a single ovariole, or they've got thousands of ovarioles, or they have exactly the same number of ovarioles every time, and ask what the developmental basis of that behavior is. And from there, we can discover the different genetic bases for controlling different ovariole numbers in these different lineages. All right, so to wrap up, the big takeaways that I have found really useful over these years from this approach is to understand that these complex adult fitness traits can ultimately, through a developmental biologist lens, come down to local cell-cell decisions during early developmental stages. There's no single smoking gun gene or regulatory pathway that can explain all of the variation in this trait, let alone its regulation in a single organism. In terms of cell-cell signaling, clearly extremely important, and every single evolved conserved animal signaling pathway has some skin in this game, and when it comes to ovarial number in this particular trait. We found this interesting link, this nutrient sensor link, this insulin signaling link between the heritable variation in ovarial number and the environmentally regulated variation in ovarial number. And this is quite exciting because it's sort of a long-standing problem in evolutionary biology to understand whether or not non-heritable variation, sort of environmentally induced variation, is relevant to the evolutionary process. Because if it's induced by the environment and it can't be inherited, then maybe you don't care about it in evolution. But if that plasticity is mediated by a pathway whose activity itself can be different in heritable ways, maybe this is a way that you can link plasticity to heredity. And then finally, we find it really fruitful to begin to think more and more about sort of null expectations for evolutionary change. We know there's going to be change, but we want to know how much change we should expect so we can know when we should be surprised and which leads we should follow next. So finally, of course, I need to thank all of our collaborators and our funding sources and the great group of interdisciplinary scientists that I work with in my lab. And thank you very much for your attention and for bearing with me as we had a couple of technical glitches. And I would just love to take any questions that you might have. Thanks.
Cassandra, that was a wonderful talk. Thank you very much. I've seen Cassandra before, you haven't seen me before. I'm Astrid Haase, I'm a tenure track at NLDDK, and I have the privilege to do the Q&A with you today. I already have a lot of questions, but for everyone who has a question, please use the send live feedback button. I think it's on the bottom of your screen, and just send us your questions and we will ask Cassandra. I would also like to repeat the CME code for today, which is 37934, 37934. Okay, thank you very much. With that, Cassandra, we could start the questions. Can I start with perhaps something simple? In addition to nutrient sensing, do you see any relationship with population size?
Ooh, that's so interesting. So effective population size measurements for Drosophila are pretty garbage. But to sort of spitball this, I think that we find that species with lower effective population sizes are going to have lower variance in ovarial number. I don't know that we can predict a lot about their absolute ovarial number or their mean ovarial number, but I think that their variance is going to be lower. I mean, that's sort of you might guess that anyway, because we know that high degrees of evolvability come with different population sizes. And so we might expect lower variance in a population with a lower effective population size. But that would be my prediction. That would be my prediction. And based on the small amount of empirical data that we have, I think that may be the case.
Yeah, that's super cool. There's one question from Susan Harbison, and she's wondering about your simulated organism, which is super cool, and really wondering about how do you want to integrate this in your research? How do you choose the characteristics?
Yeah, that's such a great question. So, the simulated organism that we've worked with the most is actually a cricket embryo. And so we were doing this sort of relatively unrelated study looking at early cell movement decisions in insect embryos. So those of you who study insects probably know that the first divisions of insect embryos aren't holoblastic as in vertebrates. So the first mitotic cleavages skip cytokinesis and you end up with one cell with a lot of nuclei inside it because they replicated and they separated, but they didn't put down an active contractile ring and they didn't separate into separate cells. So you make this bag of nuclei, then the nuclei will have to move to populate the cortex, then they get cell membranes, they cellularize, and now you have a blastoderm. Looks just like any other animal embryo, really. Okay, so the question is, how do the nuclei go to the right place? Because if they're all just born in the middle of the yolk and they just hang out there, you're never going to make an embryo. They have to move out. Even in Drosophila melanogaster, we don't know the mechanisms that drive that. So we studied this problem in a way bigger embryo that develops much more slowly, that has a nice transparent eggshell, and we can see it through its cricket. And we made some movies of the cricket and we quantified the nuclear things. And then we developed a mathematical model that was really good at explaining the behavior of these nuclei. The mathematical model predicted that a specific parameter, which was actually nuclear density, was a key driver of all the behaviors and the movement and the mitotic behaviors in the system. And then we did a combination of sort of real experiments and in silico experiments where we made embryos. We could make them in the computer because you could make anything in a computer that had specific nuclear densities or specific sizes and shapes or specific perturbations that would change the density or change other parameters that we cared about. And then you say, okay, if the nuclei behave according to this rule, let's simulate what it would do if I did this, if it did that. And then in the lab, you can do this and you can do that. And that way you can see to what extent your model is really a good predictor of the real world. And so that paper was just accepted today in Nature Communications. It's on bioRxiv, so you can read it if you're interested. But going forward, I want to take the sort of simulated organism work in at least two directions. One is more insect eggs. So we recently did another study where we sort of quantified the morphospace of insect eggs. What are the sizes and shapes of insect eggs? Turns out there's a huge amount of variation in shape and size. And we hypothesize that the nuclear movement will be some function of the shape and size of the egg itself. And then in the context of what I was just talking about here with ovarial number, you have this ball of cells, the larval ovary, a lot of cell movements, directional cell movements, polarized cell shape changes, intercalation behaviors have to happen in a specific way for you to end up with a specific number of terminal filaments. And so, when I started this work years ago, I actually started by thinking, let's make some models. And then I quickly realized we have no parameters to put into the model. We don't know how many cells there are. We don't know how often they divide. We don't know where they go. But now we know those things. So now I think it could be fruitful for us to build a simulated developing larval ovary and subject it in silico to different environmental, mechanical, genetic, cell behavioral changes. Observe what the ovarial number output is. Let's say you pick that as an output. Then we can observe whether or not real ovaries subjected to those same sorts of changes behave in the same way. And that'll help us know sort of how far off are we in knowing all the different parameters that are important to ultimately determine the right number.
Could you make hybrids of some of these species and then actually look what co-segregates and what is the dominant trait?
100%. And so, you know, as Susan well knows, this is a quantitative trait, highly variable among closely related species, including those that can interbreed. It's prime territory for QTL analysis. And indeed, many researchers over the years have done QTL analysis on this trait. And they have uncovered, I would say the QTL analysis and interspecies crosses that have been done over the last century have told us three main things. One, it's a complex trait. A lot of genes are involved. It's not Mendelian. There's not a single locus. Two, it's not sex-linked. So it's autosomal, which basically means like, you know, 2 chromosomes in Drosophila melanogaster. And three, the QTL analyses that have been done so far have uncovered, you know, a number of loci of predicted large effect, but the total number of genes contained in all these loci is numbering in the thousands because the loci were very large, because the maps were very poor resolution. It's been a good 10 years since anybody did an ovarial number QTL. And I think that the maps are such high resolution now, it would be a great trait to try to map now with today's tools. But one thing I can tell you is that we took a look at some of those older QTL studies and we picked genes with high LOD scores, so predicted to have perhaps a high degree of variation at the sequence level that explains the variation in the trait. So high and low LOD scores, and we tested them functionally, systematically, maybe 300 or 400 of these we tested by knocking them down in Drosophila melanogaster to ask what the potential functional relevance of these QTL suggested hits might be. And the answer was something like 2/3 of the loci that the QTL analyses had uncovered. When you knock them down in melanogaster, you'll change the ovarial number. But there was no relationship with LOD score, right? But I really attribute that to the very poor resolution of the maps that were available at the time. And so I think the time would be good now to try to repeat that.
So that would be super cool.
Mm-hmm.
Yeah, it would be really cool if we could the technology is much better now, isn't it? If we
Absolutely, absolutely. And, you know, also relevant to this, and people have done this as well, it's not too hard to select, to experimentally evolve a ovarial number. And so you can select, Lee Engstrom was able in the 70s to within 10 generations, select for significantly lower or higher ovarial numbers. But when he stopped applying selective pressure, both extremes snap back to that mean. And so this tells us that there's heritability in there, there's standing variation, you can select it up and down. But
it is also revertible. You can revert it.
You can, yeah, you can. You can quite easily. You basically, all you have to do is stop selecting and then it'll snap back.
So that really means that there is a certain optimum and you can transiently deviate, but then it takes a bit to constantly deviate.
That's it. I think maybe perhaps related to the species specific sort of variances. There are, you know, species specific degrees of standing variation, genetic variation that can push it up and down. But you can't really transform, not that we or others have seen yet, you can't transform a melanogaster number into, you know, one of these dung beetle numbers of 1,000. But it would be great, it would be interesting to try. Yeah.
Cassandra, Chris is sensing me that we should come to an end and give you a break before you meet with the students.
Okay.
So thank you again so much for the talk. That was amazing. And I guess everyone enjoyed it as much as I did. So thank you very much from all of us. And it will be also available on videocast for all the students who haven't been able to watch it live. Thank you very much. It was wonderful meeting you.
Thank you so much, everybody. Bye.
Bye.